Okay, good morning. Today we are going to deal with the muscular system. So the muscular system will be in two lectures. This lecture will be about the histology or the microscopic anatomy of the muscular system, and then the first lecture next week will be about the gross anatomy of the muscular system. So we will deal with the muscular tissue or the microscopic anatomy of the muscular system. There are three types of muscle fibers in the body, and these are the skeletal muscle, the cardiac muscle, and smooth muscle. So the skeletal muscle, which you can see them here, they are called skeletal because many of them, or most of them, are attached to bone. So they are attached to the skeleton, so they are skeletal, although some of them are not only attached to bone, but they are attached to fascia, or they are attached to the skin. Like, for example, the muscles that move the facial, that move the skin of the face and called muscles of facial expression, they are on one side attached to a bone, but on the other side they are attached to the skin, and so they can move the skin. But yet many other muscles are attached between two bones, and so they can move the one bone at a joint. So they are called skeletal muscle, and they are striated muscles, and we will see what is the meaning of striation. Striation here means that there are light and dark bands in the muscle, and these are very clear when you see them under the microscope. Not all the muscle fibers are striated, as you will see that there is smooth muscle fibers, for example, which are called smooth because they are not striated, but some muscles are striated, like the skeletal muscle. The skeletal muscle is not only striated and attached to the skeleton, but it has another feature, is that it is under voluntary control. So we can initiate the contraction of this muscle by our will. It is not involuntary. For example, the cardiac muscle is involuntary. It can contract, it has its own rhythmicity, and at the same time, the increase in the heart rate or the decrease in the heart rate is not under our voluntary control. The same thing is true for the smooth muscle. They are involuntary muscle. They have their own rhythmicity of contraction, and the certain hormones and the autonomic nervous system controls the contraction of these muscles. They are not under our voluntary control, but the skeletal muscle are under voluntary control. So they are under voluntary control, they are striated, and they are generally attached to bone, and so they are called skeletal. The cardiac muscle fiber here, they are striated. You can see the cross striation here, but they are involuntary. The other feature of these muscles is that they are branching. If you compare them with the skeletal muscles are in the form of cylinders. The muscle cell, I'm not talking about the muscle as a whole, but as the muscle cell. The cell is cylindrical and is non-branching, while you can see here that the cardiac muscle is also cylindrical, but it is branching. And the other feature of the cardiac muscle is that the cells are connected by what we call intercalated discs. So these are the intercalated discs here. You can see them even under light microscope. They are seen as dark lines connecting between these muscles. The intercalated discs are sites of junctional complexes between the muscle fibers, between the cardiac muscle cells. Remember when we talked about the junction between cells in general, we mentioned about tight junctions, we mentioned about gap junctions, desmosomes, hemidesmosomes, zonal adherence junctions. So there are many junctional types between the cells. Here we have two types of junctional types. There are gap junctions and there are desmosomes. The desmosomes will connect the cardiac muscles together, the cardiac muscle cells together, And the gap junctions will allow communication between these cells. So the cells together, they act as a network because there are gaps communicating signals between the cells, and the cardiac muscle cells, they act as a network or what they call syncytium or network because of the presence of these gap junctions. The gap junctions and desmosomes are present at spaces, at places which are called intercalated discs. So the intercalated disc is a junctional complex formed of desmosomes and gap junctions. These muscles, again, these are under involuntary control. So they are, although they are striated like the skeletal muscle, but they are under involuntary control. You cannot control the contraction or the rhythm of your heart. It is involuntary. And as I mentioned, that they are autorhythmic. They have what we call a pacemaker, a built-in pacemaker. They can contract and relax at a certain rhythm. But for example, when there is the autonomic nervous system can override this and can increase the rhythmicity. Like for example, when you when there is sympathetic overstimulation, when somebody is afraid, or when you are running, for example, the autonomic nervous system involuntarily increases the rate and force of contraction of these muscle fibers, and when you are resting, the muscle fibers contraction will be, the rate will be reduced. So it is not under our voluntary control, and in addition to that, there is an autorhythmicity in the contraction. The other type of muscle fibers in the body are the smooth muscle fibers. And these smooth muscle fibers, they have contractile elements inside them, but the contractile elements, they are not arranged so that they produce the dark and light bands that are present in the skeletal muscle or the cardiac muscle. So when you look at them under the microscope, they are not striated, and so they are called smooth muscles. Usually the muscles are small, the muscle cell is small, and as you can see it here, when it is relaxed, it is a spindle in shape. And when the muscle contracts, these smooth muscle, when they contract, they will not only shorten, but they will twist around themselves like a helix. And again, they are involuntary. So they are involuntary, same as cardiac muscle, but they are smooth, unlike the cardiac muscle. Cardiac muscle are striated, same as skeletal muscle, but they are involuntary, unlike skeletal muscle. Okay? Now, examples of these smooth muscle fibers, we have seen them in the skin, for example, the erector pylorus muscle that is connected to the hair follicle and causes the goosebumps in the skin. And also they are present in many other places in the body. They are present in the walls of hollow organs, like in the wall of the urinary bladder. They are present in the wall of the gastrointestinal tract and the respiratory passages, and the wall of the uterus, and so on. So they are present in so many places in the walls of so many organs in the body. The skeletal muscles, if we go back to the skeletal muscle here, you will find that there is another feature of skeletal muscle, is that the muscle cell, I'm always talking about skeletal muscle as a cell, not as a, because we are talking about the histology today. So as a cell, the skeletal muscle is multinucleated. It has multiple nuclei. And as you can see here, that these nuclei are present at the periphery of the muscle fiber, of the muscle cell. If you compare it with cardiac muscle, for example, you will see that the cardiac muscle, usually it has a single nucleus, sometimes two nuclei, but usually it's a single nucleus. Same is true for the smooth muscle. And the nuclei here for the cardiac muscle or the smooth muscle, they are centrally located nuclei. But the nuclei for the skeletal muscles are located in the periphery, and they are multiple. The reason for the multiplicity of the nuclei in the skeletal muscle is embryological. So these muscles, the skeletal muscles, they develop from embryonic connective tissue. Remember the mesenchyme that we mentioned before, the embryonic connective tissue? So they develop from mesenchymal cells that will produce myoblasts. The myoblasts that divide and produce skeletal muscle fibers. But here, the myoblasts will join together. So a single muscle fiber, mature muscle fiber, is formed maybe of 100 myoblasts fusing together. That's why it will have 100 nuclei. So they are multinucleated, multiple nuclei. Tens of nuclei are present inside them because they are formed of fusion of embryonic blast cells, myoblasts. Some of these myoblasts, they retain their ability to divide and regenerate, and they are attached or closely related to the mature muscle fiber and are called satellite cells. So these satellite cells, which are very closely related to the muscle fiber, they will help in a limited regeneration of the muscle fiber. So they can repair the skeletal muscle fiber to a certain extent, but they cannot divide and replace a lot of muscle fibers. So when skeletal muscle fibers are destroyed for one reason or another, then there is a limited regeneration and repair of these muscle fibers, and then the remaining muscle fibers will be replaced by fibrous tissue. So the muscle fiber will become weak because the fibrous tissue is non-contractile. It's unlike the muscle fiber which has the feature of contractility. Fibrous tissue contains collagen fibers and elastic fibers. It doesn't contain contractile elements, as we will see in a moment, that are present in a skeletal muscle. The same is true for the cardiac muscle. Cardiac muscles, they cannot divide and regenerate. So when the heart is affected by disease, for example, like in myocardial infarction, where there is a reduction in blood supply of the cardiac muscle that results in death of cardiac muscle, then these cardiac muscle fibers, they cannot regenerate, and this will result in replacement by fibrous tissue or scar formation. But cardiac muscle and skeletal muscle fibers, on the other hand, they can hypertrophy. Hypertrophy means that they increase in size. So when they increase in size, it means that the organelles in the cytoplasm will multiply and increase, like the contractile elements, the actin and myosin fibers, which I am going to talk about in a moment. They will have more sarcoplasmic reticulum, they will have more mitochondria, so the single cell will increase in size. So hypertrophy of the muscle, when a muscle increased in size, a skeletal muscle or a cardiac muscle, it means that the cell increased in size, but the number of muscle cells which are present is not increasing, because the muscle fibers, they cannot multiply. They can increase in size, but they cannot multiply. This is not true for smooth muscle. Smooth muscle, it can increase in size, so it can undergo hypertrophy. And on the other hand, smooth muscles can increase in number. They still retain their ability to multiply. And this is called hyperplasia. So what happens, for example, in a gravid uterus, let's say that the uterus has smooth muscle fibers in its wall, and during pregnancy, the uterus increases in size, a huge increase in size in the uterus, and thickness in its wall. This thickness is not only produced by increase in the size of the smooth muscle fibers, but increase in the number of smooth muscle fibers. This is not true for a skeletal or a cardiac muscle. The function of the muscle tissue is that evidently it produces movements of the body, and not only it produces movement, but it maintains the position of the body. So now when you are sitting, by effect of gravity and forces applied to your body, then your head will fall forward. If it were not for the muscles of the back of the neck that prevent your head from falling forward. Now, if I am standing still, not moving, if I am standing still, then the forces applied to the body will cause my body to fall forward. But there are muscles, especially the muscles of the calf, they are contracting to produce a tone that prevents my fall. So muscles, when they, some of the muscles, they contract and produce movements, but there's another type of contraction of a muscle that does not produce movement, but just creates a tone in the muscle that prevents a fall. So now if you are sleeping, then your head will fall, okay? But otherwise, you are unaware of the tone of the muscle. So the skeletal muscles not only produce movement, but also they maintain body position and posture. Also, muscles like smooth muscle fibers, for example, or cardiac muscle fibers are used for storage of substances within the body. Not only storage, but they propel substances. Like, for example, blood, which is present in the blood vessels. There is smooth muscle fibers in the wall of blood vessels. Lymph, urine in the urinary bladder, where there is smooth muscle fibers in the wall. Air in the respiratory passages. Food and fluids in the GI tract. So that, like, for example, in the stomach, it stores some of the food and fluid, and then when the muscles contract, the smooth muscles contract, they will propel that food from one place to another, or prevent it, because some of them will contract at sphincters, so they will prevent the passage from one place to another. And also the muscles, they produce heat. When the muscles contract, they produce heat, and this controls the temperature of the body. That's why, for example, when we exercise, we use the term, for example, warm up, because when we are exercising, we are actually warming, we are producing heat. So that's why this term is used. And sometimes the muscles, the skeletal muscles, involuntarily contract in order to maintain the body temperature, and this results in shivering. So that the muscle fibers involuntarily contract, this will produce heat, and this happens in cold weather. This represents the role of muscle in maintaining the temperature of the body. The muscle tissue has certain properties that are not present in many other tissues, like it is excitable. So it is excitable, that means that it responds to signals, it responds to electrical stimuli that are conducted by nerve cells. And in this respect, it might also mimic the nervous tissue, as we will talk about it next week. But at the same time, they are contractile. So the contractility, that's what I mentioned previously. I said that when the muscle is damaged, it will be replaced by fibrous tissue, and this will reduce the efficiency of the muscle, because fibrous tissue is non-contractile. The muscle tissue is contractile. It can shorten and generate force. Also, they can extend, so extensibility, and they have elasticity. So when they are shortened or when they are extended, they can spring back, because they are elastic, they can spring back to their original shape. Now, I will deal with more details of skeletal muscle. As you can see here, that this is the part of the muscle that we call the belly of the muscle, or the flesh of the muscle, and this will give rise to a tendon. The tendon is formed of connective tissue. It's not muscle tissue. It's formed of dense, regular connective tissue. It appears white because it doesn't have blood supply. The tendons, they don't have blood supply or a good blood supply. But the muscle usually, or the flesh of the muscle, appears red because there are a lot of blood vessels supplying the muscle, and at the same time they have inside the muscle, there's a special type of protein that attracts oxygen, and this is called myoglobin. So that's why the muscle fiber appears red in color. And the muscle as a whole, as you can see here, this is a cross-section of the thigh, for example, and you can see the muscles, these are groups of muscles. So the muscle as a whole is surrounded by connective tissue, which is dense, irregular connective tissue. We call it epimysium. And then each muscle consists of fascicles of muscle fibers or muscle cells. So this is a muscle fiber, muscle fiber, muscle fiber, or cells. Together, they form fascicles. And these fascicles are surrounded by connective tissue, which is called perimysium. So we have epimysium, that is the fibrous envelope of the entire muscle, and then fascicles of muscle fibers are surrounded by connective tissue, which is called perimysium, and then each cell, each muscle fiber is again surrounded by fibrous tissue that is called endomysium. And this fiber, these layers or these envelopes, they are not layers, these envelopes of fibrous tissue are important because they, first of all, they allow the passage of nerves and vessels that supply the muscle. Nerves are important for the excitability of the muscle, and the blood vessels are important to supply the necessary oxygen for contraction. And at the same time, these connective tissue fibers are going to collect at the end of the muscle, at the sides of the muscle, to form the tendon of the muscle, and they will transmit the strength of contraction of the muscle to the bone or to the skin or to wherever the muscle is attached. So three envelopes: endo, peri, and epimysium. This is again to show you a muscle fiber. So this is a muscle fiber, which is a muscle cell, and the group of muscle fibers, they constitute a fascicle. So no, sorry, this is an entire muscle fiber. I'm sorry for that. So this is an entire muscle fiber, and you can see that it is long, it is cylindrical. There are multiple nuclei. For example, here's a nucleus, here's a nucleus, and this is another nucleus. And these nuclei are located at the periphery of the cell. The cell membrane has a special name here. It's called the sarcolemma. And the cytoplasm here has a special name. It's called the sarcoplasm. And you can see that the sarcoplasm is filled with these threads of myofibrils and myoglobin. So these are called myofibrils, and each myofibril consists of filaments. So myofibril consisting of multiple filaments. These multiple filaments here, they are the contractile proteins in addition to the myoglobin. Now let's see what happens in a muscle fiber. So again, this is a muscle fiber. It has a sarcoplasm, and which is the cytoplasm, and a sarcolemma, which is the cell membrane. And there are filaments, there are myofibrils inside it, and each myofibril, this is one myofibril, it consists of filaments. Two types of filaments: thin filaments and thick filaments. So the thick filaments are called the myosin, and the thin filaments are called the actin. You can see here, this is the myosin filament. For each myosin filament, there are two actin filaments, 1, 2, and then 1, 2, 1, 2. So two actin filaments for each myosin filament. And these filaments together will constitute a myofibril. A group of myofibrils will constitute the muscle fiber. In addition to that, you can see here that there is an extensive network of smooth endoplasmic reticulum remember the endoplasmic reticulum, which is one of the organelles of the cell. There's a rough endoplasmic and smooth endoplasmic reticulum. Here we have an extensive network of smooth endoplasmic reticulum that no ribosomes are attached to it, and these act as storage for calcium ions. Calcium ions are very important for the contraction of muscle fibers. In addition to that, the sarcoplasm, the cell membrane, the sarcolemma, or the cell membrane of the cell, it has invaginations in the form of tubules inside the cell. So there are, you can see here that the sarcoplasm, the blue part, is invaginated inside the cell. And you can expect that these invaginations here, they contain the interstitial fluid. And so the action potential or the electricity that will be transmitted across the cell membrane can easily diffuse and rapidly diffuse to include the entire muscle fiber. So the entire muscle fiber will contract at the same time because of the presence of these what we call transverse tubules or T-tubules, which are invaginations of the cell membrane. I will repeat this in a moment. So, as I mentioned, that there is a network of very rich network of tubular sacs, which is the smooth endoplasmic reticulum that contains calcium ions, and whenever there is a signal, an electrical signal, then the endoplasmic reticulum will release the calcium into the cytoplasm. And this calcium will act in such a way that it will result in what we call cross bridges between the thick filament and the thin filament. So the thick filament, if you look closely at the thick filament, you will see that the thick filaments, they look like the golf clubs. They have heads and they have handles. So the heads of the golf clubs will, because when there's an influx of calcium, the head will attach to the thin filament, and it will change its shape in such a way that it will flex and will pull the thin filament in. And here again, the thin filament will be pulled in, and this results in muscular contraction. Okay? So that's why the calcium is very important. Calcium results in the formation of cross bridges between the thick and thick filaments. When these cross bridges are formed, this results in approximation of the thin filaments in between the thick filaments. And this is what we call the sliding theory of muscle contraction. Again, I will repeat it in a moment. I'm going back to the T-tubules, the invaginations of the cell membrane, the sarcolemma. You can see here, the sarcolemma is invaginated and forms tubules inside the cell, inside the muscle cell. These skeletal muscle cells are large cells. They are very large cells. And many of them are very long. You can imagine like a muscle in the lower limb, like maybe 30 or 35 centimeter in length. Each muscle fiber, each cell is very long. So there are, the cell membrane dips inside the cell, and by this way, the action potential, the electrical signal, will be distributed uniformly in the muscle fiber. This action potential is necessary for the release of calcium. Calcium is released from the smooth endoplasmic reticulum. Once calcium is released into the cytoplasm, then the cross bridges will be formed and the muscle contracts. Okay? So also you can see that inside the muscle there are a lot of mitochondria. So there are many mitochondria arranged along these fibrils, these myofibrils, and these are necessary to produce ATP, because once the cross bridge is formed, then it should be released again. Otherwise, the muscle will continue contracting like this. So the release of this cross bridge requires ATP. So that's why we need a lot of mitochondria here to provide the energy that is required for the release of the cross bridge. Okay. So for example, what happens after death? There is a condition which is called rigor mortis, which is rigidity after death. This is because of the after death, the calcium will be released from the endoplasmic reticulum, and this results in the formation of cross bridges between the thick and thin filaments. So the muscle contracts, but there is no ATP. The person is dead. There is no ATP. He is not able to generate ATP. So these cross bridges will continue, and the muscle continues as rigid, contracting, and becomes rigid. And the condition will continue for about 24 hours until the enzymes, the hydrolytic enzymes, will be released from the lysosomes and will destroy everything, including the muscle, including these cross bridges. But at the beginning, because the calcium is released from the smooth endoplasmic reticulum, cross bridges are formed, no ATP, so the muscle continues in its contraction, and this results in rigor mortis. So going back again here to the concept of the sliding filament theory of muscle contraction. You can see here, this is the thick filament, and here is the thin filament. And the thick filament, clearly it looks like a golf club. It has a head and it has a long arm here. When calcium is released, it will attach itself to the thin filament, the actin filament, and will bend, and so it will bring the thin filaments in, and this results in shortening of the muscle. Now here you can see that there's a region in the muscle fiber, or the muscle in the fibril here. There's a region where it contains thick filaments, and so this region will appear dark, while the region that has only thin filaments will appear light. So there are dark regions, light regions, dark regions, and light regions, and this results in this striation. That's why these muscles are called striated muscles, okay? Because of this arrangement of thick and thin filaments. At the same time, you can see that the thin filaments are attached to what we call, it's a kind of structural protein which constitutes a Z line or Z disc. And here is another Z disc. Between one Z disc and another Z disc, the region is called a sarcomere. So this is a sarcomere, and when the muscle contracts, the thin filaments will go in and the Z lines will be approximated. So you can just imagine that when the muscle contracts, the length of the muscle fiber shortens, but these filaments, the thick and thin filaments, they are not changing in length. They are just sliding in between each other, but they don't change in length. So when the thin filament here slides in, it will cause the Z line to be approximated here. And on the other side, when this thin filament goes in, the other Z line will be approximated to this direction. And so the sarcomere as a whole shortens. But the filament itself is not shortening. The filaments are sliding in between each other, and this will result in shortening of the entire muscle fiber. Okay? Again, I repeat that this shortening of the sarcomere is brought by the attachment of the thick filament heads to the thin filaments, and this takes place when there is calcium ions. Calcium ions are present in the sarcoplasmic reticulum. They are released when there is a nerve impulse. Okay? And in order for these heads to be detached from the thin filaments, you require energy. If there's no energy, there's no ATP, they will not detach. Rigor mortis. So this is the sliding filament mechanism of contraction. Okay, you can see here when the muscle contracts, this is a relaxed muscle, and that is the distance between two Z lines, which is the sarcomere. But here, when the muscle contracts, you see that the Z lines are approximated to each other. The length of the thick filament and thin filament is not changing. What happens is that the thin and thick filaments, they are sliding in between each other, as you can see here. Okay. I mentioned about nerve and blood supply of the muscle, and the nerve supply of the muscle is a motor nerve supply of skeletal muscle. I'm talking about the skeletal muscle. So you can see here, this is the axon of the motor neuron. We're going to talk about this next time with the nervous tissue. And when it reaches a muscle fiber, it will become distended, and what we call it an end bulb. End bulb. And in the end bulb, you see that there are multiple vesicles. These are secretory vesicles. Remember the secretory vesicles when we talked about the cytology? And these secretory vesicles, they contain a chemical called acetylcholine. When there is an electrical signal in the nerve, then these vesicles, secretory vesicles, will attach to the cell membrane of the end bulb of the nerve and will release this chemical, the acetylcholine, which acts as a neurotransmitter. So you can see here that the nerve is not directly attached to the muscle. There is a gap, and this is the region of a synapse. Okay? So there is a junctional gap here, and within the gap, this gap will be filled with acetylcholine when it is released from the nerve. This acetylcholine acts as a nerve transmitter, will be attached to the wall of the muscle of the muscle fiber and will result in an action potential or an electrical signal in the muscle fiber. This electrical signal releases calcium, and calcium results in contraction of the muscle. Yes?
What's the difference between acetylcholine and a different neurotransmitter? Because you just said that acetylcholine wasn't a neurotransmitter.
Oh, no, the others are present in other junctions. I'm talking about a neuromuscular junction, because the other neurotransmitters are present between neurons, different neurons. But at the neuromuscular junction here of the skeletal muscle, it's acetylcholine that is used. Okay, so that's why there are certain substances. For example, a material which is extracted from plants in South America, which is called curare. And this competes with acetylcholine on the transmitter. So acetylcholine, even if acetylcholine is released, it will not cause contraction of the muscle. And the muscle, therefore, will not be able to contract, will be paralyzed. And that's why they use it, for example, in game hunting, because it causes paralysis of the animal when it hits the animal. Another example of structure of substances or toxins that affect the neuromuscular junction is for example, the Botox, which is now used for removing, for example, the wrinkles from the skin. It is injected into the muscle, and this will prevent the release of these acetylcholine vesicles, and so the muscle will be paralyzed and will not cause contraction. It will not contract, and the wrinkles will disappear, because the wrinkles are because of the contraction of these muscles. So this is another application of acting on the neuromuscular junction, Botox and curare. Now, Muscle fibers are variable, skeletal muscle fibers are variable. The variation depends on the amount of myoglobin they contain, the amount of capillaries and mitochondria. And for example, in the chicken, that will be very clear. The chicken has red muscle fibers and white muscle fibers. So because of the amount of myoglobin and capillaries, red muscle fibers are present in the leg and thigh of a chicken. The white are present in the breast. So the red muscle fibers are usually the postural muscles. They are slow fibers, but they can maintain their contraction, while the breast muscles, the white muscle fibers, which contain less myoglobin, less capillaries, they are fast twitching fibers, but they cannot maintain their contraction for a long time. So the muscle fibers in general, I'm talking now about human, each muscle has different types of muscle fibers. Not all of them are fast contracting, not all of them have a lot of myoglobin. There are variations in the types of muscle fibers, and depending on their content of myoglobin, their resistance to fatigue, depending on the way that they will extract energy either from glycolysis or from oxidative pathway. So they are variable. As you can see here, a section of a skeletal muscle showing different types of muscle fibers. Generally, there are three types of muscle fibers. There are red muscle fibers, the red slow twitch muscle fibers, and these are mainly the postural muscles that are, for example, present in the calf or in the back, which they are required for long time contraction. Or the fast twitch muscle fibers, which are usually whitish in color. They have less blood vessels, less myoglobin, and they can rapidly contract, and there are intermediate muscle fibers in between. Muscle fibers can increase in size, as I mentioned, skeletal muscle fibers, and this is called hypertrophy, or they can decrease in size, and this is wasting or atrophy. And I mentioned about rigor mortis as well, and what else we have? Anabolic steroids, you read about this. And some muscle diseases, actually, they are genetic muscle diseases, like the muscular dystrophies that usually affect males, and because they are sex-linked, they are linked to the X chromosome, and since the males have one X chromosome, that's why it appears in the male. And usually it appears in young people between the age of 5 and 12 years. In this disease, in muscular dystrophies, for example, the Duchenne muscular dystrophy, the sarcolemma, which is the plasma membrane, is easily torn during muscle contraction. So the muscles are destroyed, the muscle fibers are destroyed and replaced by fibrous tissue, and the patient will be weak and disabled, and this disability is gradually building all over the time. Okay? Any questions?